Okay, welcome back everyone. This is chapter 6, and this is the third video in chapter 6. So for the week that we were gone, that we weren't meeting in person, you needed to watch part 1 and part 2. Part 3 is what we will be discussing when we're back in class. So you're not required to watch this for the day that we are away from school, but this is a just here for you if you miss class on that Monday, or if you just want to go back and have notes of something. So we're going to talk about the cytoskeleton, the cell wall, and ways that cells combine with each other, can connect with each other. The cytoskeleton is a network of fibers that go throughout the cytoplasm, very similar to the skeleton in your body is a network of bones that goes through your body. And just like your skeleton organizes your structures and your activities, the cytoskeleton is going to organize the cell structures and activities. A lot of organelles are attached to the cytoskeleton. It gives it its shape. It kind of gives it its structure. And there are 3 types of structures in the cytoskeleton: microtubules, microfilaments, and intermediate filaments.
Watch the movement of granules containing the dark pigment melanin as they travel along microtubules in a fish epidermal cell.
So I just want to say, most of the videos in this chapter aren't things you need to take notes on and like memorize facts from. They are just to see how these things work in action. So this is just showing you these bright lines are the microtubules, and these dark spots are melanin pigments. And these granules of melanin pigments are going to travel along the microtubules. So you can see how the cell uses the microtubule as a way to move the pigments where they need to move it. And this kind of allows fish to be able to do, or this particular kind of fish is able to camouflage itself by changing its pigment. And it changes its pigment by moving its melanin around itself.
This movement of melanin granules enables the fish to change its color to avoid predators. Rhodamine-labeled tubulin was used to visualize the microtubules. Notice how some of the microtubules appear to move as tubulin monomers are added to one end and removed from the other.
Right here,
Credit, Gary Borsi, Northwestern University Medical School.
So they're pointing out here how it looks like they're moving, but they're not actually moving. It's just that tubulin is being added here, so the skeleton is growing, and tubulin is being taken away here, so it just stops showing up because your cell is breaking it down there. Okay, here's actin. So the last video was tubulin. This is actin. These are different types of cytoskeleton proteins. This is going to be actin and dendrites. Dendrites are cells of your nervous system, and they spread out in these, like forked, branched patterns. And you can see this backbone of these cells.
See how actin forms extensive branched networks in the dendrites of a neutral cell. This neuron is expressing a fusion protein consisting of green fluorescent protein linked to beta-actin, a non-muscle actin. Notice how the actin microfilaments give the dendrites their characteristic shape. Credit: Hue Zhang, Robert Wood Johnson Medical School.
Okay, I just think these are beautiful. I love to see microscopy in general, but these are just so beautiful to see. Oh, and here is the cytoskeleton, and you can actually see it almost looks like the cell is like pulsing and the cell is not moving, but you can just see these proteins, these bright spots are the cytoskeleton proteins moving.
See how actin filaments interact with microtubules to guide the movement of a neuron. Fluorescence microscopy was used to visualize the fluorescently labeled actin. Notice how the actin exhibits retrograde flow as it transports microtubules back toward the cell body at the lower left. Credit: Paul Forscher, Yale University.
Okay, so the cytoskeleton is going to help support the cell and maintain its shape. Especially things like actin will give kind of a scaffolding framework for the cell to be based around. Your cytoskeleton, especially tubulin, microtubules, will provide kind of a highway. That first video we saw the fish melanin moving around. So your microtubules can provide kind of a highway to allow motor proteins, like this motor protein, can carry vesicles to and from places where they need to go. This carrying, motoring vesicles around does take ATP, so it does take energy to move things around in it. Here's another micrograph of, this is the microtubule made of tubulin, and then these are the vesicles wandering around on these motor proteins being carried around. Okay, so this one's going to show organelles, and in vivo means it's actually in a living cell. This isn't like a fixed slide or anything.
Watch as organelles are transported along.
So these ridges are the cytoskeleton, and each of these little bumps, these are organelles that you're watching move. So you're watching the organelles, these little bumps, move along these lines.
Microtubules isolated from the giant axon of a squid cell. These images were made using video-enhanced differential interference contrast microscopy. Notice how, once ATP is added, organelle movement is observed even when the microtubules are outside the cell cytoplasm. Credit: Ronald Vail, University of California, San Francisco.
This is just lovely. These cells are just lovely. Oh, let's watch.
Watch the movement of granules.
Sorry, you watched that one. Okay, so there's three main types of fibers that make up the cytoskeleton. The first one we're going to talk about are microtubules. Microtubules are the thickest. They're this coil of tubulin proteins that form a coil similar to this. And in this image, the microtubules are stained green. So you can see how they're covering the whole cell. They help act as a maintaining the cell shape. So your book calls them compression-resisting girders. So they stop your cells from being compressed. Tubulin can be important in cilia and flagella. These are components of cells that help your cell move. They can help chromosomes move, and they can help organelles move. So those images we just saw where things were moving from one side of the cell, tubulin is kind of just this highway system that lets things move, whether it's chromosomes needing to move to opposite sides so your cell can divide, or an organelle needing to move from one side of the cell to the other. Again, microtubules are made of tubulin. Here are these functions we just talked about. Then microtubules can help move centrosomes and centrioles. These are going to be important in moving your chromosomes during mitosis. In animal cells, microtubules grow out from a centrosome near the nucleus. So there's a nice little important fact that I'm never going to ask you about. In animal cells, the centrosome has a pair of centrioles. So you'll see two centrioles together. And each of them have 9 triplets of microtubules arranged in a ring. So 9 triplets. So it's 1, 2, 3, 4, 5. If you go around, there's 9 of those. And they're each in a triplet. So it's 1, 2, 3 in this one. 1, 2, 3 here. 1, 2, 3 here. You can kind of see on here. This one's the micrograph's a little bit harder to see. But these are still 9 sets of triplets. So 9 sets of these 3 little dots that make a ring. Cilia and flagella are made of microtubules, or their movement is controlled by, the movement of microtubules controls the movement of flagella and cilia. So here you have flagella on probably sperm cells, it looks like, and they're going to just beat up and down, kind of like a fish swimming. They're going until the sperm moves this way. Cilia are going to move more like oars on a boat. You do have cilia on cells in your body. So like your upper respiratory tract has cilia lining like your trachea. So that as you are breathing, if you get a piece of dust or something, if you breathe it in, or if you breathe the bacteria, it's going to get stuck in the cilia, and then you can just cough it up. So that cilia will kind of move things up if they are not supposed, so they don't end up down in your lungs. So this is a flagella. These are a bunch of flagella on bull sperm. And you're going to see this one get cut off. But what's really interesting is that the flagella can still move because it still has an ATP source. And this video does recycle through as she's narrating it. So don't think that they were like repeatedly cutting off a flagella or something. It's one little clip that's just repeating itself.
See how a flagellum beats in the presence of ATP, even when separated from the cell. The flagellum of one of these bull sperm cells was cut in half using a glass microprobe. Notice how the excised piece of the flagellum continues to beat in the absence
Totally
of
cut
any
off.
connection to the original cell.
Still beating.
Credit: Dana L. Holcomb-Weigel, Kathleen L. Leish, and Charles B. Linderman, Oakland University.
Okay. So this is talking about what cilia and flagella are made of. Never mind. Hopefully we get that to play in class. Okay, the next one we have are microfilaments. Microfilaments, sometimes you'll see them called actin filaments. Actin's pretty thin. It's two little strands wound around each other. I love actin. I think actin is so beautiful. It is a tension-bearing element. It can help hold out the shape of the cell, help stretch it out. It can change the shape of the cell. So if actin is rearranged, it can rearrange the shape of the cell. This also helps with muscle contractions. So cells need to be able to expand or constrict for muscle movement. Actin is also important in cytoplasmic streaming in plant cells. So cytoplasmic streaming just means the way that things move through the cytoplasm. It's important in cell motility and amoeboid movement and division of animal cells. It is found in bundles of microfilaments, and microfilaments can make up microvilli. So this is probably an epithelial cell, maybe lining like your intestinal tract or something. Instead of just having one solid lining for the food to just go over and you grab what nutrients you can as it moves through your intestines, instead they're in these microvilli. And so your food moving through your intestines can actually fill in all of these gaps. So you have a lot more surface area to absorb nutrients out of your food. In your respiratory system, it gives you more surface area to absorb oxygen out of the air you've breathed in. Microfilaments can function in cell motility. They'll also often have the protein myosin along with actin. Cells are able to crawl along a surface by extending pseudopodia. So this picture of an amoeba or some cell doing amoeboid movement, you can see it reaches out a pseudopod here. Then that pseudopod can stick and it kind of just like pulls itself up. So these pseudopodia are extending forward. So the cell is going to pull itself in that movement. Once it's pulled itself up, it can release from here, and then this will pull forward into the next pseudopod. This will pull forward and make a pseudopod. So it just kind of lets a cell grab on and just like cling on and pull itself up. Here's a video of cytoplasmic streaming. So you can see things moving around in this cell. You can see these things moving around in this cell. Each of these outlines shows you the cell wall of these, whatever plant cells they are. And you can see the movement is staying within a cell. It's not moving from one cell to another. So it's just flowing through the cytoplasm of one cell. That is called cytoplasmic streaming, and that is because of actin. The third filaments or the third component of their cytoskeleton are intermediate filaments. So intermediate tells you kind of in the middle. So they're not as thin as those actin fibers. They're not as thick as those microtubules. They're intermediate. They're still involved in maintaining cell shape. They're able to hold tension as they stretch. They help anchor the nucleus and other organelles, and they help form the nuclear lamina. I hope you remember that phrase. That is a protein layer around the nuclear envelope. So this table is just going through a synopsis of each of those functions that I've lost my words. This table is going through those 3 different types of cytoskeleton components. I'm just kind of giving you a synopsis of each one. Okay, next, let's move on to outside the cell. In plants, you have a cell wall. The cell wall is extracellular. It's outside of cells. And this is a feature that distinguishes plant cells from animal cells, because plant cells have cell walls and animal cells do not. We also see cell walls in prokaryotes, so bacteria with their peptidic glycan, or archaea with their pseudomurine, fungi with their chitin. Some unicellular eukaryotes, so we talked about how some protists can have a cell wall. And the cell wall will help protect the cell. It is just, a physical barrier, so things can't just come up and damage the cell. It helps maintain its shape because it's a little bit thick and inflexible, which kind of helps, if it keeps the shape, that's kind of the shape the cell has to take. And it prevents excessive uptake of water. So if you imagine a water balloon that is just all by itself, you fill it with water and you keep forcing water in it, eventually it's going to pop. But if you have a water balloon that has a thick wall around it, as you keep adding water, eventually you're going to get to a point where that thick wall around it is going to push the water back out. It's going to give enough pressure to push the water back out. And that's the same thing that cell walls do. If they are in a hypotonic environment, which is not a word you know yet, but if they're in an environment where water starts flowing into the cell wall eventually is going to apply pressure to not let that water flow on in. Plant cells are made of cellulose, if you remember that from our discussion of biomolecules. And in those cellulose, you'll have other polysaccharides and proteins embedded within that cell wall. Plant cells have multiple layers. One is the primary cell wall. In here, you can see the primary cell wall as this is a primary cell wall for this cell, and this is a primary cell wall for that cell. It's pretty thin and flexible. There are middle lamella. The middle lamella is what's here, and this is a thin layer between the primary walls of adjacent cells. So to keep this primary wall and this primary wall separate, this middle lamella is in the middle. It's between them. And then in some cells, we'll have a secondary cell wall. And the secondary cell wall is added between the plasma membrane and the primary cell wall. You can see here that secondary cell wall can be pretty thick. Oops, sorry. In animal cells, although we don't have a cell wall, we do often have a sugary layer around our cells, which is called the extracellular matrix. The extracellular matrix looks something like this. It's not a thick supporting structure. It's relatively thinner, but it can be a little bit elaborate. You can see it's got a lot of different things in it. So here we've got collagen, we've got fibronectin, we've got polysaccharides and carbohydrates all around here. So collagen, proteoglycans. Proteoglycans means proteins with sugars on them, and fibronectin. And these are often bound to proteins called integrins. Integrins are proteins that go all the way through the plasma membrane of a cell and are involved in things like adhesion and signaling between different cells. The extracellular matrix helps support the cell. It helps adhere both to a substrate or some substance you want it to stick to, or it can help cells stick to each other. It can help the cell move. So, if it's not sticking to another cell, it can kind of help it, kick off and push against it. It also can help regulate what is going on in the cell. Then let's talk about connecting cells to each other. So in things like animals and plants, we are made of a lot of cells, and those cells need to be able to connect and coordinate and work together. So we need to adhere, interact, and communicate. And these often happen through direct physical contact. There's 4 types of intercellular junctions: plasmodesmata, which we're going to see only in plants, and then tight junctions, desmosomes, and gap junctions, which we could see in our own cells and animal cells. Plasmodesmata are channels that go through plant cell walls. So this is one cell wall of one plant. This is the cell wall of another plant. And these are plasmodesmata that go straight through. And this will allow water and other small little solids, so just small little things dissolved, small sugars or ions, sometimes even proteins and RNA can go from one cell to another cell. Then in epithelial tissues of a person, you can see these three types of junctions. Tight junctions, so this is a tight junction here. These are important for membranes to press them together to prevent the leakage of fluid. So if you imagine this is an epithelial tissue lining your intestines. So you've got your intestinal fluid, you can think of what that is, flowing through your intestines, flowing through your bowels. You don't want that leaking out of your intestines and just floating through your body. So these tight junctions keep your epithelial cells stuck very tightly. So things that are supposed to be on one layer stay on that side. Then we have desmosomes. Desmosomes are, where's my pointer? Desmosomes are like this. Sometimes desmosomes are called anchoring junctions because they anchor cells together. They make them be fastened into strong sheets. So if you think of like a beating heart, those cells have to be able to move together. You don't want those cells letting go of each other. They need to work together. Or if you think of when you're moving your cells, cells need to be fastened together, and those connections need to be strong so that when your muscle moves or your heart beats, it doesn't just throw off a cell. And then gap junctions. Gap junctions are sometimes called communicating junctions because they let your cells communicate with each other. So they're channels that let things actually move between from one cell to another. Here's a picture of our video of desmosomes. Hopefully that will play in class. Gap junctions and tight junctions. Sorry, we'll see if we can play those in class. Okay, then this lecture ends with a summary of your different cell structures. This is provided for you. This whole lecture PowerPoint is in your D2L page. So I'm not going to read through these, but please make sure you are comfortable being able to tell me what a nucleus, the structure and function of a nucleus, of ribosomes, smooth endoplasmic reticulum, rough endoplasmic reticulum, Golgi apparatus, lysosomes, vacuoles, mitochondria, chloroplasts, and peroxisomes. Those are each organelles that you need to be very comfortable with. This is a very important chapter, so you need to be very comfortable being able to tell me about those organelles. Okay, I look forward to seeing you all again, and hope you're having a good week. Write to me if you have any questions. Bye.